Okay, so why don't we just go ahead and get started again while a few people trickle back in. Okay, so we just talked about optogenetics, chemogenetics for modulating neural activity. We're going to move on and talk about contemporary approaches for resolving anatomy between cell types in the brain. So we'll talk about a few different approaches. And then the last major thing we'll go through is ways to actually study the encoding properties of circuits and cell types and sort of ways we can do that. Okay, so one of the sort of bonuses that you can get from doing optogenetic manipulations is I mentioned that in addition to expressing opsin proteins such as channel rhodopsin within cells, we usually have a fluorescent protein that's conjugated to the opsin that we're interested in. So this is usually a fusion protein between channel rhodopsin and yellow fluorescent protein, for example. And so what this allows us to do is to not only manipulate the cell types like we just talked about, but you can actually visualize where these cells are projecting to in the brain. So this gives us a way to essentially screen the entire brain to see where fluorescent fibers originating from your cell type of interest are ending up in the brain. So you can think of this as sort of analogous to classical anterograde anatomical tract tracing studies with the added advantage of this being done in a cell type specific fashion. So with classical anterograde tracing, you would typically inject a tracer into a region and it would label every cell in that region and where it projects to. With this anterograde viral tracing strategy, we're starting with a particular subtype of cell and then looking at where they're projecting in the brain. And so, yeah, this is basically just what I said. You have to wait some amount of time for these fluorescent proteins to be trafficked down the length of the axon to presynaptic fibers. In the mouse brain, typically we wait anywhere between 4 to 8 weeks for full trafficking throughout all the fibers. And then the idea is that once you've labeled all these fibers from the cell type that you're interested in, is that you go in and you quantify the fluorescence in all the brain regions that you're interested in. And this sort of gives you an indirect measure of the degree of innervation from the cell type that you're targeting to where they're projecting to. And additionally, it's helpful if this is followed up with functional manipulation. So it's nice to be able to visualize where these cells are projecting to or any given cell type is projecting to, but if you can follow this up with sort of slice electrophysiology or other approaches to assess functional connectivity, I think it really strengthens what you can learn from these approaches. Okay, so that's sort of forward tracing or anterograde tracing. There's also novel approaches that have been developed for retrograde tracing of genetically defined circuits. So probably the most widely used approach for this now is based on a genetically engineered version of the rabies virus. And so what's unique and interesting about rabies virus is it's a viral, it's a virus that essentially infects synaptic terminals and then is propagated back to the cell bodies of neurons. And so this has been engineered, this whole system has been engineered by a number of labs, such as Ed Callaway's lab, who really started with the development of this approach, but has been carried on by a number of other labs now, to use rabies virus as a way to trace the number of inputs to a defined set of postsynaptic neurons. And so I just wanted to quickly go through how this works. And it requires a number of different viral constructs and tools to do this. Essentially, you're going to be starting with a set of neurons in a brain region that you're interested in that express Cre recombinase. So for example, we can start with tyrosine hydroxylase expressing neurons, just going back to our example, in the ventral tegmental area or substantia nigra pars compacta. that are expressing Cre recombinase. And the rabies virus has been, in this case that's used for retrograde transsynaptic tracing or monosynaptic tracing, has been engineered in a way where it can't actually infect neurons unless it has a receptor that's required for a rabies virus to enter the cell. And so this is this TVA receptor, which is the receptor that essentially is required for the rabies virus entry. So you have an adeno-associated viral construct that expresses this rabies receptor in a Cre-inducible manner. So you could inject this into the starter cell. Now this allows rabies virus to infect those starter cells. In order for rabies virus to replicate and propagate retrograde across synapses, there's one other protein you need, which is this rabies glycoprotein. So this is a protein that's required for the replication of the virus. If this gene is not expressed in the cell, the virus can't replicate and propagate. And so the version of rabies virus that's used for tracing isn't making this. In the normal sort of wild type rabies virus, the genome would have would express this protein and it would just propagate indefinitely. But in this case, it's been engineered where this protein's missing, and we're going to be delivering it through another virus to allow it to propagate. So in the starter neurons, or the cells that are often referred to as starter neurons, these are the cells that are expressing Cre recombinase. You express both the TVA, the receptor for virus entry, and the glycoprotein. And so you typically wait a couple weeks after injecting these constructs. You then come back in and you inject your G-deleted rabies virus that is going to express a fluorescent protein of interest. And so what this permits us to do is to first only infect the starter neurons that you're interested in looking at their inputs. And so they'll be the first to express the GFP that's encoded in the rabies virus. But then very rapidly, over the course of a few days, the viral particles that are produced by these cells will then be propagated transsynaptically into the neurons that are projecting to those Cre-positive neurons. And so why this is really cool and a potentially powerful approach is it allows you to map all those cells, or at least a large portion of those cells that are projecting to a genetically defined population of postsynaptic neurons. Does that make sense? And essentially, because the rabies virus was missing that glycoprotein that is responsible for replication or is needed for replication of the virus, none of these cells upstream have the glycoprotein. So the replication stops one synapse upstream from the starter cell population. So essentially, you just get labeling of monosynaptic inputs without labeling polysynaptic inputs. I think there's some important caveats and interpretations of these data that are important to point out. One is that typically the way this viral system is used, it's very toxic to neurons. So it's not currently it's not really appropriate to use it for functional manipulations or long-term manipulations of cell types. So you really wouldn't want to use like optogenetics to study the functional connectivity between cells with this, because typically what happens is that once the cells start expressing the rabies virus, within a short period of time, typically 7 to 10 days, the cells will die. And so it's good for looking at anatomical connections, but it's not good for long-term functional manipulations of neurons. Another important caveat is that propagation of the virus transsynaptically may have certain biases associated with it. So what I mean by that is that we don't know yet whether there's some sort of preference in terms of what synapses the virus is propagated through. So we know it works quite well for tight synapses like excitatory synapses onto neurons, things like that, but not all synapses and connections between neurons are the same. So for example, with neuromodulators such as dopamine, we know that the release sites for dopamine, like in the striatum, are sort of these loose appositions that are just close to cells, but are not necessarily always like, right there touching the cells. And so it seems, reading some of these papers, is that rabies virus isn't propagating as efficiently through those types of synaptic or neural connections as it is through like really tight pre-post excitatory synapses.
Yeah. Do you think that there will be development of a G-deleted rabies virus that you can use in the future for functional manipulation?
Yeah, there was actually a study that was just published a few months ago where they actually already engineered rabies to be less toxic. And what they showed is that the cells are healthy and viable for at least many weeks, which is a huge improvement over what you can currently do with the sort of standard one. So it's a rapidly developing technology, and I think, over time, we'll start to see more stable levels that aren't really killing cells, things like that, yeah. Okay, so this is just an example of where this was actually applied to study connections onto given cell types in a genetically defined way. This is a paper that was published by Uchida et al. In 2012 in Neuron. And they were looking at monosynaptic inputs into dopamine cells in the midbrain as well as non-dopamine cells. So what they did is to target these helper, so I should also mention that these, the TVA and the glycoprotein are sometimes collectively referred to as helper viruses for rabies. So if I use that terminology, that's what I'm talking about. So they inject these helper viruses into the VTA of a Cre-expressing animal in, I think it was a DAC mouse. And then 14 days later, they inject the G-deleted rabies virus. And then they, after another week, they take all the tissues, section it, and look at the location of the neurons. And so they noticed that by doing this, depending on what starter cell type you're looking at, whether it's dopamine neurons or GABAergic neurons that are located in the midbrain, you can see very different patterns of inputs to those cells, which is, shouldn't be too surprising since we know they're different cell types and they have different functions. But the point is that this is a method that allows us to start to look at the diversity of inputs to different cells that can be oftentimes very close in proximity to one another. Since we're talking about sort of novel anatomical approaches to resolve circuit function, the last one that I want to mention is the sort of emergence of tissue clearing approaches that really allow you to start to look at neural circuitry in the intact brain. And so sort of classically, we've been limited to look at cell types and connections in very thin brain slices. So you obviously, probably everybody here, most of us have done this before. You label some cells, perfuse the animal, take very thin slices, typically 40 microns or something, serially through the brain, and then you look at all these. The problem is that slicing the brain is sort of obviously is disrupting these connections, so then it makes it hard to actually trace where things are going to and things like that. So what's been developed in a number of different labs, sort of using competing and slightly different technologies, is ways to take opaque tissue such as the brain and essentially clear the lipids out, which then, but leave the protein structure largely intact, which then makes the brain, or what's left of the brain after you take out the lipids, relatively transparent. And so once it's transparent, you can image essentially in the intact brain, or what's left. So there's a number of different ways that you can do this, but one that's sort of becoming the current state of the art is light sheet microscopy, which allows you to take this cleared brain, which contains cells that may be expressing a fluorescent protein you're interested in, and then rapidly imaging the entire brain. And this allows you to really look at the connectivity between cells with high resolution, so we're talking cellular, subcellular resolution, but also be able to visualize in space how these cells and connections are really interacting with each other. To some extent, you can also visualize these using other imaging approaches like two-photon microscopy, but light sheet is much faster and rapidly becoming the method of choice for this. So there's been a number of papers that have been published on this over the last few years, and it seems to, every other week, there seems to be a new tissue clearing approach that comes out. But this is just an example of a recent paper that was published last week in Cell, where they essentially labeled different projection cells in the medial prefrontal cortex and looked at some of the anatomical connectivity between this structure and its projection targets. I'm not going to go into details, but this just sort of gives you an idea of what the data looks like and sort of what you can, what kind of information you can obtain from this. So essentially, some of these cells were labeled in the prefrontal cortex, and you can see their projections to a number of known targets, such as the nucleus accumbens, basolateral amygdala, VTA, habenula. You can sort of take the raw data and then graphically render it to really start to look at these different projections in a high degree of detail. And one thing that was really interesting in this paper in particular, what they did, which I think was really cool, was they actually used this approach to look at cells that were activated by a particular stimulus. So they had an animal that would express fluorescent protein only in cells that are expressing CFOS. So CFOS is a mediated early gene, is a marker, indirect marker of neural activity, and they used this to label cells that would be activated following either foot shock treatment or cocaine exposure in different groups of animals, and allow them to map where those cells that were activated by those different stimuli were projecting, and this allows you to sort of compare the differences between, say, those shock-activated cells versus cocaine-activated cells. This is just a movie from the supplement of that paper, sort of to give you an idea of what this looks like and how these things can be sort of rendered on a computer to really look at some of the connections and projections with intact tissue. So again, the important thing to stress is that this gives you cellular resolution and oftentimes the ability to trace the axons from individual cells without actually doing too much disruption to the actual tissue. Okay, so that's all I'm going to talk about for sort of the circuit architecture / anatomy stuff. I want to move on to talk about ways to resolve activity in cells. So is there any questions on the anatomy methods and what those tell you over previous things or how they can be used? Okay. Yeah.
I really like the fact that there's tracing all the way to the medial prefrontal cortex. Mm-hmm. So I wonder if what type of investigations are focusing on that region that are enabled now with the methodology that can help translation to humans.
Right. So, I mean, you can think about, so there are ways to actually trace anatomical connections in the intact human brain with like diffusion tensor imaging and things like that, right? Which actually has like quite good resolution if you set it up right and do the experiments the right way. Where this becomes analogous and I think complementary to that is that, well, first of all, the resolution's even better with like these tissue clearing approaches, right? Because we have like cellular resolution as opposed to sort of like macro scale, like fiber resolution in the brain. The other major advantage, and what's complementary to like things like DTI, is that we can do this with cell type specificity. So we can just trace the projections from, say, one particular subtype of prefrontal cortex neuron versus looking at the connections going everywhere from the cells that you would typically get with like DTI.
So it's possible to do backward translation, in other words, to look at a circuit in humans and then to say, okay, let's try to do this in mice and
right, and more mechanistically dissect yeah. So I think that's a really important point in that. Yeah, but so I think it's an important point because you know, this is a potential area where people that are doing human neuroimaging can really meet and have some common ground with people doing really nitty-gritty circuit manipulation in mice or rodents or whatever, right? Because it's very easy for us to sort of go in and say, okay, well, let's just see where this cell type's projecting or whatever. And hopefully there's going to be some commonalities with the more macro scale connectivity data that's coming out from all the human imaging stuff. Okay, so basically the last thing I want to talk about before we break into the groups for the discussion questions is ways that we can target cells to resolve their activity. So I really think of this as sort of the other side of the coin with respect to optogenetics and chemogenetics. With optogenetics and chemogenetics, you're functionally manipulating neural circuits to see what happens to behavior. But if we don't actually know the endogenous activity properties of these cells or circuits, it really makes it difficult to make informed manipulations. Like if we don't know when the cells are firing or when they're active, under what context, what's the point of doing all these different stimulation parameters? Really, it's really it's difficult to sort of put it in the context of natural behavior of the circuits unless we know how the cells are normally adapting and responding to a variety of different stimuli. And so the first way to do this is coming back to in vivo electrophysiology, which I mentioned is sort of a staple approach to really understanding how neurons are encoding activity. However, this has been adapted to be used with optogenetics and other methods to essentially give a higher degree of specificity in terms of what cells are being recorded. So what we talked about in the beginning was that you can drop electrodes in and sort of record activity from a variety of different cell types. Problem being is you don't actually know what cell type you're recording from most of the time. But you can at least classify cells based on their functional properties, for example. So what people have done recently is to put channel rhodopsin into neurons, and instead of using it as a way to manipulate neurons to see effects on behavior, you can use channel rhodopsin as an indirect marker of a cell type you're recording from. So essentially you can target a cell population to express channel rhodopsin. You can then implant electrodes that are coupled to a light source, like a fiber optic, and then apply brief pulses of light to activate those cells and detect on your recording electrodes what cells are actually being activated. So the basic premise of this, or the rationale, is that cells that are directly responding to the light pulse would be the cell type you're interested in recording, right? So like if you did this in the VTA, for example, we know that there's dopamine cells, GABA cells, other types of neurons. If you target channel rhodopsin to the dopamine cells and you pulse light, cells that show a very rapid response or a light-evoked action potential could be inferred to be dopamine cells because of that. Does that make sense? Okay, so this allows us to then phototag or identify which cells are a particular genetic subtype. You can also do this to look at projection-specific neurons. So we know that in most brain regions, you have projections to brain region A, B, C, D, whatever. Cells project all over the place. Oftentimes, it's important to try to understand whether a particular cell type, based on their projection, may be encoding information differently versus cells that might be projecting to a different brain region. And so how you can do this is, again, using the sort of twist on phototagging, is to target all of these cells with channel rhodopsin and then put an optical fiber in the postsynaptic area that you're interested in. And what you can do is that when you activate these fibers that are innervating the area, oftentimes what you'll see is antidromic activation or a back-propagating action potential, which can then be detected at your recording electrode. And so this allows you to screen neurons based on their projections to known brain regions. And so the idea is that cells that are responsive to this antidromic activation can be indirectly inferred to be projecting to that brain region where the light source is. However, if you don't see responses in cells, that doesn't necessarily tell you that they're not projecting there. They might be channel rhodopsin negative or maybe not expressing enough, something like that. So phototagging is good for identifying a subset of cells. One of the problems being, though, is that it's still relatively a low throughput approach for identifying and recording from different cell types. So this is just an example of when this was used. This is probably one of the most, well cited or described papers, again, focusing on these different cell types in the VTA, so obviously a region of the brain that's has clear relevance to addiction. And this was by Jeremiah Cohen and now Uchida, published a few years ago. And so again, they used phototagging to indirectly identify neurons based on where they express channel rhodopsin. So you can see in vivo, in this particular neuron that was recorded, you can see every single light pulse is resulting in a detectable extracellular recorded action potential. And so if this was done in targeting the dopamine cells, which it was in this paper, you would infer that this cell that's being recorded is indeed a dopamine neuron. And so using this and comparing the firing patterns between dopamine cells and GABAergic cells in the VTA, they were able to show that dopamine neurons show a very distinct pattern of activity to cues that predict rewards as well as to rewards themselves, very similar to what's been described in primates and other work where these were really just identified as putative dopamine neurons. Couldn't really definitively say that they were dopamine cells, but this gives us a much more definitive way to say, like, okay, these are for sure dopamine cells because they're we only targeted the opsins to those cells. And you can see that the firing patterns in those cells that were unequivocally non-dopamine cells or GABAergic neurons shows a very high, is very different or distinct from the dopamine cells. So just a couple things to point out is that because this is an indirect method of being able to encode or to study the encoding properties of particular cell types, there's a number of different sort of rigorous analytical and physiological controls that are needed. You have to make sure that you really are detecting action potentials from the cells you're interested in, and you're not seeing some sort of polysynaptic circuit effect where you might be stimulating one cell but then very quickly seeing a response on a neighboring cell that you might be recording at. So it still is an indirect approach for really resolving cell types. And as I mentioned, sort of the limitations is that the yield can be very low with this approach. Most of the cells that are recorded are not going to be phototagged. In addition, you can't really track cells over many days or many recording sessions. So you can typically study population activities within a single task, but if you really want to track how the activity of neurons is changing like day to day, or with learning, or with like drug exposure over time, things like that, it's not really a good method for that. So what is much better at tracking cells over time, as well as giving you a high degree of directly targeting cells, is using genetically encoded fluorescent proteins that can be used to report neural activity. So if optogenetics is typically focused upon using light-activated proteins to modulate neural activity, there's a whole other set of proteins that can be used to sort of report neural activity. And this is a rapidly developing field, and new sensors are coming out all the time that report different things, whether it's calcium influx into neurons, voltage fluctuations, protein-protein interactions, whatever. But the general idea is that you can introduce genetically encoded proteins into certain cell types that would then report neural activity, and then using a variety of different in vivo imaging approaches, you can resolve how these cells are changing their behavior in the intact brain within the context of a behavioral task. So right now there's really two different approaches that are sort of used for this. One being in vivo 2-photon calcium imaging, which we'll talk a few minutes about, which really allow you to resolve the activity of cells, say if they're expressing GCAMP, which is a genetically encoded calcium indicator. And we'll talk sort of about the strengths of this, but this typically has to be done in a head-fixed animal that would be trained on a particular behavioral task but cannot freely move. The other approach is to use these miniaturized epifluorescent microscopes that are sort of been shrunken down to the point where even a mouse can sort of wear it on its head and it'd be interfaced with the mouse, and the animal can freely move and behave, and you can record the activity of cells. So the idea here is that you have this very small microendoscope that's implanted into brain tissue that gives you optical access to cells that are expressing your activity indicator, such as GCAMP. And then you have this microscope that interfaces with this relay lens or the grin lens that you can then record the activity of these cells. So since I've mentioned GCAMP, and it's currently the standard in the field for measuring activity, I just wanted to talk about it in a minute. So this is, as I mentioned, it's a genetically encoded calcium indicator that changes its fluorescent state based on whether the protein is bound to calcium or not. So it's a fusion molecule between the calcium binding domain of CAM kinase and green fluorescent protein. And the general idea is that when it binds calcium, this causes a conformation change in the protein, which causes its fluorescence to change. And from extensive work from the people who have developed these molecules over time, as well as many other labs in the field, it's been shown that these changes in the fluorescent state of the protein correlate very nicely with neural activity. So what you can see here in this trace from the paper that published GCAMP6, which is sort of now the standard protein that most people in the field are using, you can see a trace where they're showing GCAMP fluorescent changes in a cell where they were actually recording the electrical activity as well. And so what you can see, even with single action potentials, you can see fluctuations or deviations in the fluorescence from baseline. And if you have cells or action potentials that occur in quick succession to each other, if a cell is like burst firing, for example, or a number of action potentials occur simultaneously, you see a larger increase in the fluorescence. So it seems that these proteins are accurately reporting neural activity in terms of action potentials by indirectly measuring fluorescence associated with calcium influx to the cell, which occurs during action potentials. That being said, there's obviously times where calcium may come into cells or may be changing intracellularly that might be associated with something else other than action potential. So it's still an indirect measure of the activity of the cells. It's very highly correlated with the action potentials and firing properties of the cells, but there might be other instances where calcium may be released or modulated within the cell that would still give you changes in fluorescence. So that's just one caveat. This is a movie showing you sort of what some of these data look like. So it's a little bit hard to see, but essentially what you're seeing is a very large field of view with 2-photon imaging and many individual neurons showing changes in their fluorescent dynamics associated with their neural activity changes. And so, as I mentioned, one of the major advantages of this is, well, first of all, the ease of targeting a cell type, because we can just use Cree driver lines or whatever. You know, as long as your Cree line is specific for the cell type you're interested in, it's very easy just to target and image only those cells you're interested in. The other thing is the sheer number of cells you can image. So whereas photo tagging and electrophysiology is relatively low throughput, maybe get a handful of cells per animal, it's very easy with imaging to get hundreds to thousands of cells in a single animal. So that's very powerful. The other advantage is that it's it's possible to actually record and track the activity dynamics of individual neurons over many, many sessions. So not only can you look at the activity in one particular behavior, but you can sort of track to see how those activity changes might be changing as a function of time, things like that. So this is just a couple examples to run through for that. So in our lab, we published a paper a few years ago where, or I guess last year, where we used the miniature epifluorescent microscope to look at activity, again, in the lateral hypothalamus. So we started by targeting the calcium indicator, GCAMP6, to just to GABAergic neurons within this nucleus. So again, I've already mentioned that there's lots of different cell types, and so we were, in this case, interested in what are the encoding properties of these GABAergic neurons first, and then this allowed us to eventually go on and look at other cell types as well. So we use virus, target these GABAergic cells in a VGAT Cree mouse line, and then we implant these microendoscopic lenses directly above the lateral hypothalamus and interface the miniature epifluorescent microscope to resolve the activity of these cells. And so then we can record video showing the fluorescence of these cells in a variety of different behavioral paradigms, and then we can use automated methods to extract out the fluorescent time series data from individual neurons over time. And these are just giving you some sort of representative example cells that we saw. Just to give you an idea of sort of what the data looks like, at least in this deep brain region of the hypothalamus. This is a recording from an animal that was doing a progressive ratio schedule of reinforcement where they had to nose poke a number of times to earn a reward. In this case, it was palatable sucrose reward. And so you can see from this animal that's freely moving, these individual sort of flickers of fluorescence represent activity from individual neurons that were recorded in the awakened behaving animal. So we can use this to then look at how these activities of these cells, of these what we know are now GABAergic neurons, how are they changing with respect to different behavioral parameters. And so the two things that we looked at was two different very easy to discriminate behavioral events. The consumption of the reward, so when the animal actually goes to lick and consume the sucrose, versus how their activity is changing during the chain of nose pokes that they have to make to earn the reward. So you can look at sort of activity time lock to the action of nose poking or the action of consuming the sucrose. And what we saw was that in both cases, we saw a number of cells that showed increases in their activity time locked to these different events, the appetitive nose poke events or the consummatory lick events. What's really interesting about this approach, and one of the other strengths, is that it allows us to sort of compare how individual cells are responding to these different events. So what I'm showing you here is the activity of this field of cells that we were recording from, time-locked to the consumption of the reward or to the nose poke responses. And so what we saw was that individual neurons that were responsive to the consummatory events when the animal was consuming the sucrose tended to not respond to the nose pokes, and vice versa. So when the cells, if we see cells that are responsive to the nose poke events, we see that they're not really responsive to the consumption of sucrose. And so this really allows us to start picking apart the encoding properties of cells, but in a genetically defined context, since we were just recording from one subpopulation of neurons. And then finally, just to talk and show you some example data from 2-photon imaging. This is data from the lab where we've been recording from neurons within the prefrontal cortex based on their projection targeting, and Jim actually will be presenting a poster on this work here, so if you want to see more about it. And what we've been doing is to use head-fixed behavioral tasks to look at how neurons in the prefrontal cortex, based on their projections to downstream target regions, such as the nucleus accumbens or thalamus, VTA, et cetera, how are they differentially encoding reward predictive stimuli. So we have tasks where animals that are head-fixed and under the 2-photon microscope can use a cue to guide their behavior. So we have a cue that's associated with a reward, a conditioned stimulus, that's always followed by sucrose, and we have another stimulus that's never followed by the delivery of the reward. And then we can probe to see how different cell types are encoding these based on the projection targets. And I'm not going into the details, but I'm just going to show you sort of representative data from this task where we can measure the activity of hundreds of sort of neurons simultaneously within this brain region, either from all prefrontal cortex neurons or from sort of cells based on their projection targets. And then we can look at the activity of large populations of cells to these sort of very simple behavioral tasks, such as the cue that predicts reward versus the cue that doesn't. And so one thing to highlight is just the sheer number of neurons that we can record from. So one of the sort of complaints about in vivo electrophysiology is that you may be undersampling the number of cells. So if you only record 50 cells, how do you really know that's accurately reflecting the dynamics of the entire population? With in vivo imaging, this is at least partially circumvented because we can record from so many cells simultaneously. So with this, I'm showing you the recordings of, I think, about 1,500 neurons that were recorded in just a handful of animals. And so this really allows us to systematically profile different populations of cells that will then be projecting to different brain regions.
Yeah. You mentioned that we can record simultaneously from different neurons. Okay. Is that possible to move the crowd and record simultaneously from different coordination or different depth, or we should just fix it in one specific coordination and just have cover?
Yeah, so it depends on the imaging approach you're using. With 2-photon imaging, you can definitely move your objective and get different depths through the tissues. So for example, if you wanted to record from different layers of the cortex, right? It's possible to, depending on where you're imaging, say image from like layer 2, 3 cells and then go a little bit deeper and get layer 4 or layer 5 cells, and then look at the differences in patterns of activity within those different layers, things like that.
So in that case, for example, while we want to investigate or see the activity in parallel cortex, we can just go to layer 4 or 5 and simultaneously record the activity.
Yeah.
In that case.
Yeah. And so that's another advantage with imaging over electrophysiology, because oftentimes with electrophysiology, it's can be very difficult to really know which layer you're recording from, and most times people don't really report that. They just report the aggregate population of what they see, probably recorded from all layers.
So for something like kind of understand how it works. So you've got like this 5 seconds. So you've got a couple of seconds to be recording. Do you record one or several neurons at the same time?
They're all recorded essentially at the same time. So what you're seeing in this field of view is essentially, so these are all individual cells that are, basically recorded from that field of view. And then you have to use sort of analytical methods to extract out the time series data from each one of those individual neurons. So there's a lot of data processing after you actually acquire the data that you have to do in order to resolve single cell activity patterns from these sort of movies that I've been showing.
The advantages. So for example, you've got CS plus, right? Mm-hmm. How many stimulus and you can still have the same activity? So
So typically we do we like to have like trial-based structures where we have like the same stimulus presented sort of randomly but over and over and over again, because then it allows us to see how robust is this response. Does it happen on every single trial versus some of the trials, things like that. So in most of the experiments in my lab that we're working on, we would typically have like 50 presentations of the CS plus and 50 of the CS minus over each recording session. And then each animal would be recorded every single day to look at how the activity of individual cells change over learning of the task and things like that. Does that make sense? Yeah. Okay, so the other major advantage, as I said, is that you can actually not only record these large populations of cells sort of on a single day, but it's very relatively straightforward to resolve the activity patterns within the exact same cells over many days. So what I'm showing here is a 2-photon projection image from a small field of cells recorded on one day, a few days later, and then a few days after that. And it's very easy, even just by eye, to pick out the exact same cells over days. So then this allows us to look at how the activity patterns of these cells are changing every day. And then finally, the last thing I wanted to touch upon is that typically 2-photon imaging has been used in the most superficial layers of the cortex, so like layers 2, 3 of the cortex, which in my opinion, it's been great for resolving activity in those areas, but obviously there's many areas of the brain that we're interested in that are much deeper in the cortex. So the question, or much deeper than the most superficial layers of the cortex. So the question is, how do we resolve activity patterns in those brain regions with these approaches? And so what we've been doing, and a number of other labs have been working on recently, is to adapt 2-photon imaging to record from deeper subcortical structures, many of which would be important in addiction, like the nucleus accumbens, ventral tegmental area, etc. And so how we've done this is to combine 2-photon imaging with an implantable grin lens. So this gives us optical access to tissue deep in the brain and allows us to resolve activity still with the nice spatial resolution that you get with 2-photon imaging. So this is just an example sort of image showing that. And I'll just end showing you some data from 3 different brain regions, 2 in the hypothalamus and 1 in the ventral tegmental area in dopamine cells, showing that we can actually resolve individual activity of cells in these deep brain regions using 2-photon imaging. So we can reliably track the activity of these cells, individual neurons, over many weeks to months in a head-fixed behaving animal. And then finally, the last thing I want to mention in terms of ways to resolve neural activity is another approach that's been being widely adopted within the field is fiber photometry, which can be used to assess bulk changes in activity within a particular cell type or projection. So this is an approach that uses fluorescence as a readout of neural activity. However, it doesn't give you cell-level resolution. You're getting a bulk fluorescent signal that is an aggregate of activity from many cells or projections from many cells. So it's essentially, you provide excitation light to activate, to excite the calcium indicator, such as GCAMP, and then, which is coupled to a fiber optic, allowing the animal to freely move. And then the emission from the fluorescent protein comes back up and hits a photodetector, which converts that light into an electrical signal, which allows you to resolve these very fast blips in fluorescence. And so again, this is a way to monitor changes in the activity of cells, but it's not at a single cell level. So it's a bulk change in the activity. So in some cases, this is fine. Like if all the cells may be acting in a very homogeneous fashion where they're all showing essentially the same patterns of activity, these approaches would work fine. But if you're working with a cell type or a circuit that there might still be a lot of heterogeneity in the response patterns, that some of that information will certainly be lost by looking at the bulk fluorescent signal seen here. Okay, so just to summarize, what we've talked about is that by really building upon these classical approaches to modulate neural circuits such as lesions, electrical stimulation, et cetera, we're really, we've recently, meaning like in the last 10 years or so, really been moving in and using modern approaches in system neuroscience to really facilitate our understanding of the precise wiring and function of the brain with a much higher level of specificity for manipulations, but also for studying the encoding properties, anatomy and connectivity between cells that really wasn't possible before. So, some things that I would like you to all think about is how in the future can we leverage these approaches to really understand the neural circuitry of addiction with a much higher degree of specificity. So, I sort of presented this in a very generic context of like, okay, here's the tools that are available and sort of what's emerging from the field right now to study brain or circuit behavior relationships. But as we move forward with this course throughout the week, I want you to think about how we could leverage these to really put it towards a particular application, such as addiction.